Good afternoon, kids. And today we will be starting with our next chapter, which happens to be chapter number 11, biotechnology. Right now, when I say biotechnology, there are two parts and they belong to unit 9. Okay. Now, when I say biotech, we will be first learning about a chapter called as principles and process. Once this chapter is done, we will be learning about the next chapter that happens to be chapter number 12. It is said to be application. Again, it is a part of biotech ka application only. Okay, and both belongs to unit 9. Now, let us first start with our basic important thing. What exactly we mean by the word biotechnology? Now, of course, everyone knows that bio is nothing but life. Okay, and when I say technology, definitely we will be talking about computers, we'll be talking about various different engineering principles. Okay, so when you apply engineering principles in biological field, then a new hybrid field has been developed, and that field is called as biotechnology. Okay, so in simple words, biotechnology is nothing but application of engineering principles in biological field. Now a question arises, why this particular application came into picture. Okay, now listen, let us take very simple example. We use engineering principles for obtaining curd. Now you may ask me how. So can I say we use a microorganism called as lactobacillus. Okay, right. Now this lactobacillus definitely converts our milk into curd. But please understand this particular conversion is done at a proper temperature. अगर आप लोगों ने देखा रहेगा तो जो कर्ड है वो विंटर सीजन में बहुत टाइम लेता है बनने के लिए, और एग्जैक्टली उसका ऑपोजिट होता है ड्यूरिंग समर. So now, if you allow that particular bacteria to grow, you will get the curd production at a very high rate. So again, can I say we have manipulated the temperature for our benefit. For example, in winter, what we do is we little bit warm, we take warm milk, okay, and then we add that inoculum. Inoculum is nothing but the हमारा जो पुराना दही रहता है उसको हम लोग उसके अंदर डालते हैं because it contains bacteria and we call it as inoculum. Okay, now I'll give you one more example. Now, kids, you must have heard about an antibiotic called as penicillin. Okay, now this penicillin is very, very powerful antibiotic. That is, this chemical kills bacteria. Okay, and it is obtained from a particular fungus called as Penicillium notatum. Now, understand this. If I want to produce this penicillin at a, I can say like in much more larger volume, I need to have big reactors, like a size of a building. उतने बड़े vessels होते हैं, right? और ये जो vessels है उनको हम लोग बोलते हैं bioreactor. अभी ये bioreactor वापस काम करता है on principles of engineering. Okay, very clear. So when you apply engineering principles in biological field to obtain goods and services. Now let me explain what I meant by that. Okay, हमने, देखो सुनो ध्यान से, हमने इंजीनियरिंग के प्रिंसिपल्स को बायोलॉजिकल फील्ड में अप्लाई किया. जैसे मैंने बोला, मैंने टेम्परेचर को कंट्रोल किया, राइट मैंने प्रेशर को कंट्रोल किया, मैंने पीएच को कंट्रोल किया, और मैंने इंडस्ट्रियल लेवल पे ये पेनिसिलिन बनाना चालू किया. गेटिंग इट? तो ये जो प्रोडक्ट है पेनिसिलिन, इसको मैं बुलाएगा गुड्स. गेटिंग माय पॉइंट? राइट. अभी आप लोगों ने शायद आपके स्कूल लेवल पे एक राइस के बारे में सुना होगा जिसका नाम है गोल्डन राइस. गोल्डन राइस इज सेड टू बी विटामिन ए रिच राइस, यानी अगर आप चावल खाएंगे तो आपको कार्बोहाइड्रेट के साथ साथ विटामिन ए भी मिलेगा. ओके, तो ये जो चावल है सुनहरा चावल, या हम लोग उसको बुलाएंगे गोल्डन राइस, इज डेफिनेटली अ प्रोडक्ट and it is supplying us with vitamin A. So definitely it also belongs to a hybrid field of biology which is called as biotechnology. Okay, so in short, we will be applying engineering principles or technology in biological field to obtain goods and services. अब सुनो, When I say biotechnology, there are two streams which we are interested in. The first is called as genetic engineering. Now, let us first understand what we mean by the word genetic engineering. So, no, the answer. Genetic engineering is nothing but it is a process by which you either add a gene or you delete a gene or you manipulate a gene. Okay. Now, let us take an example of addition of a gene. Now, let us consider that a person is suffering from diabetes mellitus. Okay. And I will say that this person is suffering from diabetes mellitus right from birth. Okay. क्योंकि उसके पास इंसुलिन बनाने वाला जीन है ही नहीं. तो मैं क्या करूंगा, मैं जेनेटिक इंजीनियरिंग का इस्तेमाल करके उसमें इंसुलिन बनाने वाला जीन ऐड कर सकता हूं. आज की तारीख में वी आर हैविंग दैट टेक्नोलॉजी. ओके, सो आई कैन क्योर डायबिटीज राइट फ्रॉम द जाइगोटिक स्टेज. ओके, यानी जब वो जाइगोट में था, मैंने उसका डीएनए बाहर निकाला, उसका एनालिसिस किया, तो पता चला कि इसमें इंसुलिन बनाने वाला जो जीन है वो है ही नहीं. तो आई विल डेफिनेटली अप्रोच जेनेटिक इंजीनियर्स and they will tell me no problem, we will do it. Okay, so what they will do, they will add a gene which will produce insulin. Okay, so point number one, genetic engineering involves addition of a gene, deletion of a gene. Now let us consider, okay, there is a disease which is called as Alzheimer's, भूलने की बीमारी. Okay, I'm not talking about dementia, I'm talking about Alzheimer's. It is a type again of a brain, I can say, disorder. इसमें एक स्पेशल प्रोटीन बनता है, मैंने ये मेरे प्रीवियस चैप्टर में भी बोला था, एंड दैट प्रोटीन स्टार्ट्स डिग्रेडिंग अवर न्यूरॉन. मुझे जाइगोटिक स्टेज में पता चलता है कि इसमें ये जेनेटिक डिसऑर्डर है, तो मैं वापस जेनेटिक इंजीनियर्स को अप्रोच करूंगा और उनको बोलूंगा कि बाबा ये देखो, ये जीन को डिलीट करना है, तो दे विल डिलीट इट. ओके, राइट. Next is manipulation of gene. Now when I use the word manipulate, okay, manipulate is nothing but जो original है उसको थोड़ा सा change करने का, ताकि effect बहुत ज़्यादा आए. For example, a person is suffering from a disease. Let us consider Sickle-Cell Disease. Okay, now listen. In Sickle-Cell Disease, I hope everyone is very familiar with a word called as codon. Okay. Now, listen, there is a codon in normal people, GAG. Now, what happens is, there is a mutation, and because of that, the codon is converted into GUG. Okay, now, this GAG codes for glutamic acid, whereas GUG codes for valine. Because of this mutation, a normal RBC, which is a biconcave RBC, is converted into a sickled RBC. Okay. Now, मेरे को पता चलता है zygotic stage में कि ये change हुआ है या mutation हुआ है, तो वापस में genetic engineer के पास जाएगा और उसको बोलेगा कि ये मेरे को problem दिख रहा है. तो वो लोग क्या करेंगे? They will replace this U with A. I repeat, they will replace the U, which was a faulty nitrogenous base, with a proper nitrogenous base. तो मैं ऐसे बोलूं क्या, हमारे इंजीनियर्स ने जो faulty gene था उसको manipulate किया into a healthy gene. Okay, so we can take care of many genetic disorders by using genetic engineering. I hope the term is very clear. We are going to either add a gene or delete a gene or manipulate a gene. Okay, or I can say that we can alter the chemistry. Alter का मतलब है change. We can alter the chemistry of our gene. That is either DNA, right? Okay, and in the case of viruses, the genetic material can be RNA. So we can change that. Is it okay? And the field is called as genetic engineering. Okay. Now, let us come to the next important field of biotechnology, which is called as bioprocess engineering. Now, what is bioprocess engineering? Bioprocess engineering is nothing but it is obtaining certain goods. Okay, now what exactly we mean by the word goods over here? Now, when I say goods in biotechnology, we are talking about vitamins. We are talking about antibiotics. We are talking about certain fermented product, okay, like alcohol. Now you may ask me, sir, alcohol, okay, so हानिकारक है, जरा भी नहीं. Of course, like when you start drinking it in excess, yes, it is. But remember, all our sanitizers, they contain alcohol, okay, right? And you know that alcohol now is a very, very good disinfectant. तो मेरे को अगर इसको इंडस्ट्रियल स्केल पे बनाने का, इंडस्ट्रियल स्केल का मतलब है बहुत ज्यादा अमाउंट में अगर मेरे को बनाने का है, then I will be requiring bioprocess engineering. Okay, I require vitamins. Again, now you are very aware, well aware that for curing COVID-19, you require vitamin C, you require vitamin D, you require vitamin E, you require proteins. Okay, right? And definitely for prevention, you require alcohol-based sanitizers. So all these products can be obtained from microorganisms, okay, and these microorganisms are processed, right, and the field is now called as bioprocess engineering. Okay, we will be dealing in detail with genetic engineering, and we will be having a brief discussion about bioprocess engineering in this particular chapter. Okay, now as I told you, genetic engineering is nothing but it is either addition, deletion, or manipulation of a gene. So I require certain tools whenever we talk about engineering. For example, okay, अगर मेरे को computers का software engineer बनना है, तो can I say my tool is nothing but my computer? In the same manner, okay, when I talk about genetic engineering, definitely we require certain tools. Okay, now my first tool for genetic engineering is nothing but we call it as desired gene. Now let us understand what exactly we mean by the word desired gene. Listen, desired gene is gene of interest. Now, what I mean by the word gene of interest, listen, now we all know that we contain around approximately 25,000 genes, okay, in a haploid cell. Now, I don't require all the genes. I require only one gene which I want to manipulate, or which I want to add, or which I want to delete. Okay, let us take for example, I want to prepare human insulin. Okay, so the gene of interest will be my gene which is going to produce insulin. And for your knowledge, you need to remember the insulin producing gene belongs to chromosome number 11. Okay, so ये मेरा gene of interest हो गया. Getting my point? Right. Okay, now my second tool in genetic engineering is very, very important, and we call them as enzymes. Now, please remember, without enzymes, genetic engineering is impossible. Okay, so my second tool is nothing but enzymes. Okay, now when I say enzymes, I have various different categories of enzymes. The first enzyme is called as lytic enzyme. Now, when I use the word lysis, lysis is nothing but breaking down. Okay. For example, if I want to break a bacterial cell, I need lysozyme. If I want to break a plant cell, I will be requiring cellulases. If I want to break a fungal cell, which is made up of chitin, I will be requiring chitinases. Okay. And for animal cell, I will be requiring a mixture of lipases and proteases. So remember, for a bacterial cell, I will be using lysozyme. Okay. For plant cell, I will be using cellulases. For fungal cell, I will be using chitinases. And for animal cells, I will be using lipases and proteases because our cell, okay, right, has got a membrane which is made up of lipoprotein. So lipid के लिए lipases होगा और proteins के लिए proteases होगा. Okay. So my first enzyme is lytic enzyme. Okay. My second enzyme is very, very important and we call them as cleaving enzymes. Now, kids, remember, when I use the word cleave, okay, cleave is nothing but to cut. I repeat, cleave is nothing but to cut. And lysis is nothing but to break. तो जो एंजाइम सेल को ब्रेक ओपन करता है वो लाइटिक है और जो एंजाइम सेल को कट नहीं कर सकते, राइट? ओके, वी नीड टू कट द डीएनए, राइट? सो cleaving enzymes are those which can cleave or cut nucleic acids. And when I say they can cut the nucleic acids, we call them as nucleases. Now, when I use the word nucleases, definitely they have the ability to cut both DNA as well as RNA because we are now focusing on nucleic acids. Is it okay? So that this is my second enzyme. My third enzyme is called as joining enzyme. Now, when I use the word joining enzyme, definitely one enzyme comes to our mind immediately, and that is nothing but DNA ligase. Now, if you remember replication of DNA, we had joined the Okazaki fragments by using DNA ligase. Okay, so ये joining enzyme हो गया. And my fifth enzyme is said to be synthesizing enzyme. This enzyme helps in synthesizing of nucleic acid. So it can be either DNA polymerase or RNA polymerases. So I will be using the word polymerases. Okay, very clear. So if I want to prepare a DNA or an RNA in vitro means outside the cell in artificial medium, I need polymerases. Okay, so these are the 4 enzymes which I am interested in. Okay, now my third requirement is said to be host. Now understand this. When I use the word host, host is that organism which is receiving the desired gene. For example, okay, I want, okay, to insert insulin producing gene in my cell. So I will be the host. So there has to be some donor and there has to be some recipient, or I will be using the word host. Okay, so host is the one which will receive the desired gene. Okay, kids, right? So host. Receive the desired gene. Okay, right. Now, there is one important tool. I will erase it, I feel, because I need to explain that in very detail, and we will be definitely dealing that in detail also later on, of course. Now, listen. My fourth tool is very, very important, and that fourth tool is called as vector. So, my fourth tool of genetic engineering is called as vector. Now, kids, if you remember a particular disease called as malaria. Now, we all know that malaria is basically caused by Plasmodium vivax. Okay, but this Plasmodium vivax, which happens to be a protozoan, okay, is carried by female Anopheles mosquito. So can I now say that that mosquito is acting as a vector to transfer that protozoan in our body? Okay, so vector का basically मतलब होता है vehicle. तो वैसे ही listen, मेरे बॉडी में अगर मुझे इंसुलिन वाला जीन डालना है तो डेफिनेटली अगेन आई रिक्वायर अ व्हीकल और आई रिक्वायर समथिंग कॉल्ड एज वेक्टर. So when I use the word vector, remember vector is nothing but vehicle. Okay, now there are different vectors which we use in genetic engineering and we have to deal with them in detail. The first vector is said to be plasmid. Okay, the second vector is said to be bacteriophage, which happens to be a virus. Okay, but remember this virus is going to only infect bacteria, therefore we use the word bacteriophage. My third vector is a hybrid vector which is called as cosmid. I will explain that, don't worry. Okay, and my fourth vector is called as plasmid. Right, so these are the four vectors which are normally used in genetic engineering. In our syllabus, we have plasmid, and I will be giving you an extra information with regards to bacteriophage. Okay, introduction to cosmid and no phasmid at all, because we are more focused on plasmid. Our NCERT covers only plasmid, but I want you to know even about bacteriophage and an introduction to cosmid. Okay, right. So these are the four tools for genetic engineering. So ये मेरे tools हो गए. Now let us come to the procedure or the methodology. Or I can also say steps involved in genetic engineering. अभी बच्चे सुनो, listen. When I say steps involved in genetic engineering, there are two methods which we basically use. The first method is called as indirect method, and the second one is called as, of course, direct method. Okay, now what is the difference between these two? Listen, indirect method is a method in which we use a vector. अगर मैंने प्लास्मिड को यूज किया या मैंने बैक्टीरियोफाज को यूज किया या मैंने कोई और वेक्टर को यूज किया तो मैं बोलूंगा कि अरे ये तो इनडायरेक्टली मैंने जीन को ट्रांसफर किया क्योंकि मैं वेक्टर यूज कर रहा हूं. ओके, एंड अनदर मेथड इज सेड टू बी डायरेक्ट मेथड और वी कॉल इट एज vectorless method. Now please understand this. Remember, when you do practically, okay, direct method is actually not advisable, because if you do 100 experiments, there is a chance of only one experiment getting successful. I'm using the word chance, वो भी नहीं होता है. Okay, तो direct method जो है वो थोड़ी सी मैं बोलूंगा flop method है as compared to indirect method. Okay, many people also call it as vector mediated gene transfer. So indirect method is also called as vector mediated gene transfer. इसका मतलब हो गया कि मैं वेक्टर के जरिए डिजायर्ड जीन को एक ऑर्गेनिज्म से दूसरे ऑर्गेनिज्म में ट्रांसफर करने वाला हूं, तो मैं उसको बुलाएगा वेक्टर मीडिएटेड जीन ट्रांसफर. Very clear? So now let's start with indirect method. Okay, now the first step in indirect method is isolation of desired gene. Now when I use the word isolation, isolation का मतलब है basically separation. Okay, तो देखते हैं कि हम ये कैसे करते हैं. सुनिए ध्यान से. समझो मैंने ये सेल लिया, लेट अस कंसीडर हमने क्रोमोसोम नंबर 11 में से सेल को सॉरी सेल में से क्रोमोसोम नंबर 11 को निकाला और उनको इंसुलिन का जीन चाहिए. तो आई विल टेक सेल फ्रॉम आइसोलेट्स ऑफ लैंगरहैंस बीटा सेल. ओके, और उसके अंदर ये डीएनए है. अभी मैं सबसे पहले क्या करूंगा, मैं इसका लाइसिस करूंगा by using lytic enzyme. As a result, the entire cell will be broken and a DNA will now be obtained. Okay, now let us consider this is my entire genome. Now you all know the meaning of the word genome. I am not interested in the entire genome, but I am interested only in a part of it. Okay, so I need to cleave the DNA. So for cleaving, I will definitely use the word cleaving enzyme. So after cleaving enzyme, I will get DNA, or rather to be very specific, my gene of interest. Okay, तो मैंने gene of interest को निकाल के साइड में रख दिया. Getting it? या मैं ऐसे बोलूंगा कि this is my insulin producing gene, जो मैंने अलग निकाल के रख दिया है. Okay, very clear kids, right? So my first step is isolation of desired gene. I am explaining you it in very brief. We will be dealing this in detail again. नहीं तो एक काम करते हैं, अभी इसको खत्म करते हैं. सुनो, listen. When I use the word lysis, if the donor cell is a plant cell, I told you we will be using cellulases, pectinases, etc. Okay. Now the moment you break open the cell, listen up. The moment you break open the cell, you will be definitely getting proteins, you will be getting carbohydrates, you will be getting lipids, etc., etc. Okay, forget about this DNA, right? So when I perform lysis, let us consider I will be getting carbohydrates, I will be getting fats, that is nothing but my lipids. Okay, I will be getting proteins, then I will be getting nucleic acid, that is my RNA and DNA. Okay, now listen, what I will be doing, I will be exposing this entire cytoplasm along with nucleoplasm, okay, to certain enzymes. For removing carbohydrates, I will be using carbohydrases. अभी सुन ध्यान से, कार्बोहाइड्रेसेस क्या करेगा, जो पॉलीमर्स है उसको तोड़ेगा, या मैं ऐसे बोलूंगा कि जो पॉलीसैकेराइड है उसको कन्वर्ट करेगा मोनोसैकेराइड में. ओके, तो मेरा अगर समझो ये बहुत बड़ा कार्बोहाइड्रेट था, तो ये इसको तोड़ेगा इनटू स्मॉल यूनिट्स व्हिच आर नाउ कॉल्ड एज मोनोमर्स. फिर मैं सेंट्रीफ्यूज करूंगा. I hope everybody now has a clear idea about centrifugation. Centrifugation is nothing but rotating or spinning at a very high speed. तो जो heavier चीजें हैं वो नीचे settle हो जाएंगी और जो lighter है वो ऊपर आएगा. तो अभी मैंने carbohydrates को तोड़ा है into monomer. So my test tube will have carbohydrates over here and lipids, proteins and nucleic acid over here, that is in the sediment. क्योंकि ये heavy है, ये नीचे सेटल हो जाएंगे और ये मोनोमर्स बहुत लाइट है, इसके लिए ऊपर रहेंगे. मैं इसको डिस्कार्ड कर दूंगा because I am not interested in carbohydrates, I am interested in DNA, rather nucleic acid. Okay, right? तो इसको मैं डिस्कार्ड कर दूंगा. तो मैंने कार्बोहाइड्रेट्स को फेंक दिया. Okay, then what I will be doing, I will be using another enzyme, and that enzyme is called as lipases. Lipases क्या करेगा, lipids को तोड़ेगा. Now I hope you know that lipid is nothing but glycerol plus fatty acid. तो ये lipases उसको तोड़ देगा. वापस मैं सेंट्रीफ्यूज करूंगा तो लिपिड का जो पार्ट है वो ऊपर आ जाएगा. आई विल डिस्कार्ड इट. ओके, तो अभी मैं ऐसे बोलूं क्या, मेरा जो सेडिमेंट है उसमें प्रोटीन्स है और न्यूक्लिक एसिड है. ओके, फिर मैं क्या करूंगा? In the third step, I will be using proteases. Proteases proteins को तोड़ेंगे into amino acid. मैं वापस सेंट्रीफ्यूज करूंगा, अमाइनो एसिड्स ऊपर रहेंगे, न्यूक्लिक एसिड नीचे रहेंगे, मैं ऊपर वाले पार्ट को डिस्कार्ड कर दूंगा. ओके, so like that we have discarded carbohydrates, we have discarded lipids, we have also discarded proteins, and now what remains at the bottom is nothing but your nucleic acid, which is nothing but DNA and RNA. ओके, अभी सुनो ध्यान से. सो अभी मेरे टेस्ट ट्यूब के अंदर डीएनए है और आरएनए है. अभी मुझे सिर्फ डीएनए चाहिए. I will be using an enzyme called as RNases. Now RNases will definitely break RNA molecule. अभी जैसे ही आरएनए टूटेगा तो उसका भी मोनोमर्स मिलेगा, मैं सेंट्रीफ्यूज करूंगा, राइट, और फिर अल्टीमेटली मुझे मिलेगा सिर्फ डीएनए. तो अभी इस टेस्ट ट्यूब के अंदर सिर्फ और सिर्फ डीएनए भरा है. ध्यान से सुनना, इस टेस्ट ट्यूब के अंदर सिर्फ और सिर्फ डीएनए भरा है. नाउ व्हाट वी विल बी डूइंग, लिसन. We need to precipitate this DNA so that we can store it. Okay. So for precipitation of DNA, we will be using chilled ethanol. Okay, we will be using chilled ethanol or ethyl alcohol. उसके वजह से ये जो टेस्ट ट्यूब है, इसके अंदर हमारा डीएनए ऐसे नूडल्स के जैसे नीचे सेटल हो जाएगा. Then what you do? You take a glass rod. I repeat. You take a glass rod, and on that glass rod, right, how we eat Maggi, in the same manner, okay, we will be wrapping our glass rod with the DNA. Okay, तो जैसे हम नूडल्स को हमारी फोर्क के ऊपर wrap करते हैं या bind करते हैं, in the similar manner, we will be wrapping the DNA on the glass rod. Okay, and this process is called as spooling. I repeat, this process is called as spooling. Now you can actually take out your NCERT book and come to page number 201. उस page number 201 पे you can see a diagram in which there is something called as spooling which has been written. Okay, so please observe that particular thing. Okay. So, if you also happen to read 11.3.1, I repeat, you have to read 11.3.1 from your NCERT book and you will be clear with what I was actually explaining. Okay, you take the cell, you perform lysis, you get everything, okay, then one by one, you will remove all the biomolecules except the DNA. The DNA is precipitated using chilled ethanol and it is collected on glass rod by a process which is called as spooling. Okay, so now I am having my DNA with me. Okay, now listen, I need to cut this particular DNA, or rather I need to take only the desired gene from this entire DNA. I am not interested in the entire genome. I repeat, I am interested only in one single particular gene or maybe a set of genes. Okay, right. So now the next enzyme which comes into picture is nothing but our cleaving enzyme. So we will now understand our cleaving enzyme. As I told you, cleavage is nothing but to cut. So cleaving enzyme is that enzyme which is going to cut the DNA. Okay, kids? So as I told you, we will definitely now be learning about cleaving enzyme. Okay, the one which will cut the DNA. Now listen, these cleaving enzymes belong to a class of enzymes called as nucleases. Now when I use the word nucleases, These are those enzymes which have the ability to cut a nucleic acid. It can be either a DNA or it can be RNA. Okay. Now, we have divided nucleases again into two categories. The first one are called as exonucleases. Now understand exonucleases. Exonucleases are those nucleotides which can, enzymes which can remove the nucleotides from the ends. That is either at the 5' end or at the 3' end. यानी अगर ये मेरा डीएनए है या आरएनए है, एक स्ट्रैंड ले लो खाली, तो अगर वो एंड से न्यूक्लियोटाइड्स को निकाल पाएगा, बीच में से नहीं कट कर सकता है, वो सिर्फ एंड से निकाल सकता है, तो उसको मैं बुलाएगा exonucleases. So exonucleases are those enzymes which can remove the nucleotides either from the 5' end or from the 3' end, but not in between. Okay. So we come to another group and we are very much interested in this, and we call them as endonucleases. Now endonucleases are those which can cut the DNA somewhere in between. यानी वो phosphodiester bond को बीच में से कट कर सकते हैं. Is this clear? Great. Okay. और इसके लिए इनको हमने नाम दिया है endonucleases. अब सुनिए, हमारी इंटरेस्ट एंडोन्यूक्लिएजेस में भी एक स्पेशल एंजाइम के ग्रुप के साथ है और उसका नाम है रेस्ट्रिक्शन एंडोन्यूक्लिएजेस. रिपीट, रेस्ट्रिक्शन एंडोन्यूक्लिएजेस, या हम लोग इसको शॉर्ट में REN भी बुलाते हैं. राइट. अब सुनिए ध्यान से. We are very much interested in these RENs because these RENs cut the DNA at a specific nucleotide sequence only. I repeat, these enzymes cut the DNA at a specific nucleotide sequence only. Let me explain exactly what I said. Listen. Now, let us consider I am having two different DNA samples, A and B. Okay. Now, let us consider I'm having a particular REN. Okay. And this REN can cleave or cut the DNA only if the DNA has got a sequence GAATTC. हिंदी में बोल रहा हूं, ध्यान से सुनना. मेरे पास एक ऐसा REN है जो DNA को कट कर सकता है only if the DNA has got this particular nucleotide sequence. अगर ये सीक्वेंस नहीं है तो वो DNA को कट नहीं करेगा. मैंने जैसे बोला, let us consider we are having two samples of DNA, A and B. Okay. Let us consider the sample contains now. You can see that there are two DNA samples I have taken, and this DNA sample contains let us take this GAA. Okay, so now this DNA sample contains this particular sequence GAATTC, whereas this sample doesn't have the sequence. Now, as this doesn't have the sequence, the REN which I'm talking about will not be able to cleave this DNA. वापस एक बार बोल रहा हूं, REN DNA को कट करेगा अगर उसके पास एक स्पेसिफिक न्यूक्लियोटाइड सीक्वेंस होगा. फॉर एग्जाम्पल, मेरे पास एक ऐसी एंजाइम है जो DNA को कट कर सकती है only if the DNA has got a sequence GAATTC. अगर ये सीक्वेंस नहीं है तो वो DNA को कट नहीं कर सकती. तो मैं बोलूंगा कि REN are very specific in their action. They cleave the DNA at a specific nucleotide sequence only. Okay, they have or a particular recognizing sequence, and if that sequence is absent, the DNA will not be cleaved. Okay, right. So this is what we are interested in, and we will be learning about REN in detail. तो चलो REN अभी पढ़ते हैं, ठीक है? तो ये शायद आपको समझा, एक्सो एंडो, एंडो में भी हमारा इंटरेस्ट रेस्ट्रिक्शन एंडोन्यूक्लिएसेस में है। ये ऐसी एंजाइम है जो डीएनए को स्पेसिफिक जगह पे कट करती है। If you are not still clear with the idea, don't worry. As the lecture proceeds, you will be definitely getting a fair idea of what exactly I am talking about. Okay, chalo. Now let us enter into the world of REN. Now kids, we were having two great scientists. उनका नाम था लिन एंड आर्बर. Okay, now listen. These Lin and Arber, these two great scientists, were studying the infection of viruses to a bacteria. If you recollect Hershey and Chase experiment, याद आ रहा है? हमने उसमें ऐसे देखा था कि एक वायरस है जिसका नाम है बैक्टीरियोफाज, और वो बैक्टीरियोफाज बैक्टीरिया को इन्फेक्ट करता है. ओके, तो उन्होंने क्या किया? उन्होंने एक बैक्टीरिया लिया जिसका नाम था ई कोलाई. गेटिंग इट? इस ई कोलाई को उन्होंने एक पेट्री डिश के ऊपर कल्चर किया. ठीक है, तो लेट अस कंसीडर ये मेरा पेट्री डिश है और ये पूरा का पूरा पेट्री डिश भरा हुआ है ई कोलाई से. ई कोलाई मेरी बैक्टीरिया है, एस्चेरिचिया कोलाई. कोलाई बोलते हैं क्योंकि ये कोलोन में पाया जाता है. ठीक है, राइट. अब सुन, उन्होंने इस ई कोलाई को इन्फेक्ट करवाया विद अ वायरस, और उस वायरस का नाम था T2 बैक्टीरियोफाज. ओके, तो मेरे पास एक वायरस है जिसका नाम है T2 बैक्टीरियोफाज, और ये वायरस इस ई कोलाई नाम के बैक्टीरिया को इन्फेक्ट करता है. अगर हम T2 बैक्टीरियोफाज को देखेंगे तो ये बैक्टीरियोफाज कुछ इस प्रकार से दिखता है. ये उसका प्रोटीन कोट होता है जो रेड कलर का मैं ड्रॉ कर रहा हूं, और उसके अंदर जो मैं ब्लैक कलर का ड्रॉ करूंगा ये उसका डीएनए है. सो दिस इज प्रोटीन एंड दिस इज डीएनए. ओके, नाउ ये बैक्टीरियल सेल है. ठीक है, बैक्टीरियोफाज बैक्टीरियल सेल को इन्फेक्ट करता है, अपना डीएनए अंदर इंजेक्ट करता है. तो दिस इज वायरल डीएनए. ओके, अब सुनिए ध्यान से. ये जो ब्लू वाला डीएनए में ड्रॉ कर रहा हूं ये बैक्टीरिया का डीएनए है. अभी हम लोगों ने ऐसे डिस्कवर किया कि ये वायरल डीएनए के पास एबिलिटी है कि वो बैक्टीरियल डीएनए के साथ अपने आप को जोड़ दे. सो we saw that the viral DNA has the ability to incorporate itself into the bacterial DNA. अभी इससे क्या होगा? इससे ये होगा कि ये जो पूरा बैक्टीरिया है उसका मेटाबॉलिक कंट्रोल वायरल डीएनए के पास आ जाएगा, और ये वायरल डीएनए धीरे धीरे बैक्टीरियल सेल को फोर्स करेगा वायरस के प्रोटीन बनाने के लिए. और कुछ समय के बाद आप देखेंगे कि इस बैक्टीरियल सेल में नए नए वायरसेस पैदा हो रहे हैं. There are new viruses which are being formed inside the bacterial cell. Is it okay? And a day will come जब भी पूरा का पूरा बैक्टीरियल सेल इन वायरसेस से भर जाएगा. तो जब ये भर जाएगा बैक्टीरियल सेल फट जाएगा, और ये सारे वायरसेस बाहर निकलेंगे। इस तरीके से वायरस का मल्टीप्लिकेशन होता है हमारे सेल के अंदर भी। अगर एक भी वायरस अंदर आया तो वो वायरस अपना डीएनए अंदर इंजेक्ट करता है, हमारे डीएनए यानी बैक्टीरियल डीएनए के साथ वो इनकॉर्पोरेट हो जाएगा और बैक्टीरियल सेल को वो फोर्स करेगा वायरस के प्रोटीन बनाने के लिए, ताकि अंदर नए नए वायरसेस बने, राइट। एक समय ऐसे आएगा कि पूरा बैक्टीरियल सेल वायरस से भर जाएगा और ये बैक्टीरियल सेल फट जाएगा, तो ये सारे वायरसेस बाहर निकलेंगे। ओके, इस तरीके से वायरस का मल्टीप्लिकेशन होता है। गेटिंग माय पॉइंट? तो ये नॉर्मल चीजें होती हैं। अभी लिन एंड आर्बर ने ऐसे देखा कि, सुनिए ध्यान से भी, लिन एंड आर्बर ने ऐसे देखा कि जब भी T2 बैक्टीरियोफाज ई कोलाई सेल को इन्फेक्ट करता है, ओके, तो ये वापस बैक्टीरिया का डीएनए है और ये जो ब्लैक वाला है वो वायरल डीएनए है। लिन एंड आर्बर ने एक बहुत अजीबोगरीब चीज देखी कि जब भी वायरस बैक्टीरिया को इन्फेक्ट करता है तो वो अपना डीएनए अंदर तो इंजेक्ट करता है, बट वो डीएनए बैक्टीरियल डीएनए के साथ इनकॉर्पोरेट नहीं हो रहा है। क्यों? क्योंकि उन्होंने ऐसे डिस्कवर किया कि ये बैक्टीरियल सेल, ओके, इस वायरल डीएनए के टुकड़े टुकड़े कर देता है। अभी जरा सोचो, अगर वायरल डीएनए के टुकड़े टुकड़े हो गए तो ये वायरस के डीएनए के टुकड़े इस बैक्टीरियल डीएनए के साथ इनकॉर्पोरेट नहीं हो सकते, और अगर वो होंगे नहीं तो डेफिनेटली नया वायरस बनेगा ही नहीं, और मैं ऐसे बोलूं क्या, बैक्टीरिया अभी भी जिंदा रहेगा। इन अदर वर्ड्स कैन आई से दैट the bacterial cell has restricted the entry of viral DNA into its own DNA. I repeat, bacterial cell somehow restricted the entry of viral DNA into its own DNA by cleaving the DNA. So, the enzyme which was successful in cleaving the DNA in between was called as endonuclease, and as it restricted, I repeat, as it restricted the incorporation of viral DNA into its own genome, right, they called this enzyme as restriction endonucleases. Is this clear? So why is it called as restriction? Because it restricts the entry of viral DNA into the bacterial DNA. It is called as endonuclease because it cleaves the DNA in between. Okay. Now, a question arises that enzyme has the ability to cleave the phosphodiester bond. अभी उसको कैसे पता चला कि ये अपना डीएनए है और ये वायरल का डीएनए है और मुझे सिर्फ वायरस के डीएनए को ही मारने का है? ओके, तो लिन एंड आर्बर ने ये सारा मैकेनिज्म वापस स्टडी किया एंड टू देयर सरप्राइज दे ऑब्जर्व दैट दिस बैक्टीरियल सेल प्रोड्यूसेस एन एंजाइम कॉल्ड एज मिथाइलेशन एंजाइम। अब ये मिथाइलेशन एंजाइम क्या करता है मालूम है? मिथाइल ग्रुप्स बनाता है और इस ग्रुप्स को अपने डीएनए के ऊपर चिपका देता है। अभी सुनो ध्यान से, ऐसे मेरा पूरा का पूरा डीएनए मिथाइल से भरा हुआ है, ओके। जब भी वो आरईएन को बनाता है, आरईएन को सिर्फ फॉस्फोडाइस्टर बॉन्ड को कट करने का है। गेटिंग इट? बिकॉज ऑफ मिथाइल ग्रुप, this REN cannot cleave the phosphodiester bond of its own DNA, okay, but it started cleaving the phosphodiester bond of the viral DNA. Is this very clear? तो पहला एंजाइम जो बना था उसका नाम था मिथाइलेशन एंजाइम और सेकेंड एंजाइम जो बना वो था REN. Is it okay? And the first REN to be discovered was from a bacteria called as Haemophilus influenza. I repeat, the name of the bacteria was Haemophilus influenza. You can come to page number 195 and you can read over there. The first restriction endonuclease discovered was Hind2. अगर आप देखेंगे तो अब ये क्या है ये मैं सिखाऊंगा. डोंट वरी अबाउट इट नाउ. अभी सिर्फ याद रखना कि the first REN to be discovered was Hind2, where H stands for Haemophilus, IN stands for influenza. Okay, and as this was the second enzyme obtained from that particular bacteria, it was called as 2. So 2 is said to be the order of discovery of enzyme from that particular bacteria. The first enzyme was methylation enzyme and the second enzyme was REN, which is Hind2. Is it okay? And one more thing, it will cleave the DNA only if the viral DNA contains GTC It will cleave the viral DNA only if the sequence is GTC, GAC. अगर ये सीक्वेंस नहीं है तो वो उस डीएनए को नहीं कट कर सकते. फॉर एग्जाम्पल सुनो, अगर आप ये सेम ई कोलाई को लैम्ब्डाफाज, ये एक दूसरा बैक्टीरियोफाज है, उससे अगर इन्फेक्ट कराएंगे तो इस सेम ई कोलाई के अंदर आपको बहुत सारे वायरसेस दिखाई देंगे, क्योंकि लैम्ब्डाफाज का जो डीएनए है, राइट, ओके, उसमें वो पर्टिकुलर सीक्वेंस नहीं है. गेटिंग इट? वेरी क्लियर. अगर वो हीमोफिलस इन्फ्लुएंजा है, ये मैंने ई कोलाई का एग्जांपल लिया है, पर जो सबसे पहला एंजाइम डिस्कवर हुआ था उसका नाम था Hind2. इज इट ओके? वैसे ई कोलाई भी इस T2 बैक्टीरियोफाज के डीएनए को मारने के लिए एक एंजाइम बनाता है जिसका नाम है EcoR1. ओके, और EcoR1 डीएनए को कट करेगा ओनली इफ द सीक्वेंस इज GAATTC. अगर ये सीक्वेंस होगा तो ही वो कट करेगा, नहीं तो नहीं करेगा. Hind2 डीएनए को कट करेगा अगर उसके पास सीक्वेंस है GTC, GAC. ओके, तो ये सीक्वेंस के अनुसार ये आर्यन काम करते हैं. Hind2 इसको कट नहीं कर सकता है, EcoR1 इसको कट नहीं कर सकता. Is this very clear? Right. So this was an introduction to REN. So when I now say REN, I will be very clear that REN are synthesized by bacterial cells when they are attacked by viruses. I repeat, RENs are synthesized by bacterial cells. We don't have the ability to produce REN. I repeat, we don't have the ability to produce REN. We produce interferons. Interferons are nothing but antiviral proteins. Okay, so we produce proteins. They produce REN. REN is different, interferons are different. Okay, so please remember this very well in your mind. So RENs are strictly produced by bacterial cells when they are attacked by viruses. Okay, and as I told you, they are very, very specific in their action. Now, let us understand nomenclature of REN. Okay, listen. Now, remember, as I told you, the RENs are named after the bacteria. For example, there is an REN called as EcoR1. Okay, now let us understand what exactly we mean by the word. Now, E, or the first alphabet, is nothing but genus. In this case, it is Escherichia. Okay, next is species, and in this case, it is coli. So Escherichia coli. R is said to be strain. Now, if you remember, I told you about rough and smooth, right? So R is nothing but strain, and in this case, the strain is RY13, and 1 is the order of discovery. So this is the first enzyme which was obtained from E. coli, therefore it is called as EcoR1. Okay. The another enzyme which you can use is Hind2, where I told you H is nothing but Haemophilus, IN is influenza. Okay, H is Haemophilus, which is nothing but genus. IN is nothing but species, which is influenza. D is nothing but the strain, and 2 is the order of discovery. So this was the second enzyme obtained from Haemophilus. If you recollect, I told you the first enzyme was methylation enzyme, and the second enzyme was this. Okay, and remember, EcoR1 will cut the DNA only if the DNA has a sequence of GAATTC. Okay, whereas it will cut the DNA only when the sequence is sorry, my mistake. Okay, GTC and GAC. Okay, getting it? Fine. So this was an introduction to our REN. In the next lecture, we will be talking more about these sequences. Okay. I hope till here you have understood. I request you to please read page number 195. Okay. So that the things would be more easier for you. Okay, kids? Chalo, bye. Meet you next time.